Okay, our second talk is from Zoom. Dr. Fulvio Gandolfi from Milan, Italy. We talk about advanced intestinal in vitro platform for the functional characterization of the feeds in aquaculture. Please. Your floor.
Thank you. Thank you. Can you hear me? Thank you. Thank you very much. Thank you very much for inviting me here. And my apologies that I cannot be there with you in presence. If you can see my slides,
No, we can't see them. You have to share them on the screen.
Sorry, let's try again.
Can you share them on the screen, please? That's good.
Does it work now?
Perfect.
Can you see them?
Yes.
Perfect. Thank you very much. Here it is. Okay. Today I'm going to talk about the results that we obtained in an European project that is just finished, and what we learned through this 5-year project, and how we are trying now to move from the pure academic application of this knowledge to a more applied environment. So the topic of the project has to do with aquaculture, and the problem that we were trying to tackle arises from the fact that there is a constant growth in aquaculture, which is driven by two factors. On one side, in the more rich countries, fish is becoming an increased commodity because of its health properties. The fish meat is considered to be an healthy source of proteins, and more in general, since its production is widespread in the world, and given the growing population, there is a growing demand of fish worldwide. As a result, it has been impossible to satisfy all this demand by the wild catch of the fish, and all the differences between the demand and the production has been covered up by aquaculture, which is a constantly growing field in animal production. There are many problems that, aquaculture is an effective way of producing animal protein, but it has a number of limitations, especially with regards to sustainability and animal welfare. Among these limitations, one of these is the source of the components of the feed. Most of aquaculture farmed fish species are carnivorous, and therefore they're used to eat. Part of their diet is made of fish feed and fish oil, and this, of course, has a limitation in the growth of the industry. Therefore, the feed industry is trying to develop more and more alternative ingredients that can substitute the marine origin component. In our project, what we tried to do was to set up an in vitro platform that could provide an efficient and a quick preliminary screen of the various possibilities. There are so many options available to the industry, different material, different sources, different combination. The cost of this material is very important because the cost of the feed in the industry covers more than half of the whole cost of producing a fish. And at present, the best way of testing the feed are in vivo trials. And of course, in vivo trials are time-consuming. They take time. They take a lot of money. And in general, as you know, there is a certain an energetic attempt to reduce the use of animal for experimental purposes. All this has increased the request and the demand of an efficient way of mimicking the intestinal function in vitro, and therefore making an in vitro intestine. Very briefly, the digestion process is grossly divided in two parts. One that we can call the luminal part, where because it happens in the lumen of the intestine, and is where the big complex molecules are broken up in smaller molecules. And then there is a mucosal part, so a parietal part, which takes place on the wall of the intestine, and that is where this small molecules are actually absorbed by the intestinal epithelium and then transferred into the bloodstream. The first part has been already developed in the previous year and is quite efficient, so it can be performed in different species, in all mammals and in fish as well. What we tried to add was the second part. We tried to mimic the intestinal wall as well as possible in a way that is as similar as possible to the real thing. In order to do that, the first step was to isolate two stable cell lines, and we have been lucky enough to being successful in this attempt. So we have a cell line derived from the proximal part of the intestine and from the distal part of the intestine, which are devoted to the first one to the absorption of the nutrients, and the second one to the process of is the site of expulsion, and is the site of the more heavy inflammatory process if something is wrong in the fish diet. So they represent the whole length of the intestine. And they are different. So they are different in vivo, and we characterize them, and they are also different. They have a number of differences in vitro as well, which, of course, is a good sign, because they maintain some properties of the original tissue, which is the main issue when you are trying to replicate in vitro an organ. In fact, as a general rule, the cells in vitro have to undergo to different demands. On one side, they are required to grow so that you can proliferate them and you keep them a constant source of these cells without going back to the animal, because otherwise it would defeat the purpose. So you cannot continually kill animals to get primary cell lines. On the other hand, a growing cell is by definition undifferentiated cells. That happens in vivo as well. And in fact, the typical cell line, so every stem cell line which grow happily in vitro, per se, when they proliferate, are stem cells. They are therefore undifferentiated. They can only differentiate it if they are correctly stimulated. And at this stage, if the cell is correctly stimulated and correctly differentiated, the cell slows down and in most cases became quiescent, as is the case of most differentiated cells in the organism. So if you look at differentiated cells, like the easiest one to look at are your skin cells. The upper layer of your skin are actually made of dead cells, so they are absolutely not dividing, but they are functional very well. They protect the inside of your body wall. They are impermeable. They don't allow water to get out. They don't allow the infection to come in, and so on. And similarly happens in the intestine. Although in the intestine is a little bit more complicated, because you don't want a complete closed barrier in the intestine epithelium, but you want the intestine to select the good things to bring in and to keep the bad things out. So the intestine is a little bit complex in this respect. So what we did in order to have the system working, so you have heard about the various options that there are available. So cell line per se are considered to be not closely mimicking the real thing, and it's actually true, as we just said. So we adopted the approach to stimulate the cell to differentiate, and to do that using what we call an enriched environment, therefore providing a more suitable environment that, as we know, has so much impact in differentiated, undifferentiated cells. As I have no time to go in all the details, but basically I will briefly illustrate the two models that we study more in detail. One is a rather simple model. Both of them, sorry, are bicameral models. So there is an upper chamber. Yeah, can you see my pointer here? Maybe here. There is an upper chamber which mimics the intestinal lumen, and the basolateral chamber which mimics the blood flow. So in the simple model, we have just a simple membrane in which we plate the epithelial cells. And then we have a more complex model, what is commercially available model, which is the Alvetex, which looks like this, is a polystyrene sponge in which the holes that you can see here at the electron microscope are of the right size in order to stimulate fibroblasts to home in these spaces, empty spaces, so that they fill the spaces, and then they do something that you normally don't see in fibroblasts grown in vitro. Usually fibroblasts are considered to be very nice epicell because they keep on proliferating, but this is very far from what the fibroblasts actually do in vivo. In vivo, fibroblasts don't proliferate that much, but just stay at some distance from one another, and they fill up the space with extracellular matrix, which is actually very important for the many things, among other, to guide the differentiation of the epithelial cell which stay on top of them. So these are the two models that we used. One, just a simple layer of epithelial cells, and the other, the same epithelial cells, but grown on this 3D support, filled up with fibroblasts, which are producing a nice extracellular matrix. And this is what happens to the fibroblast when you grow them in different ways. So if you grow them on a classical plastic surface, or in a culture flask, in a petri dish, you see the red labeling here is just collagen, so which is one of the main components of the extracellular matrix. So the collagen stay within the cells. This is not secreted, and fibroblasts are close to each other. Whereas if you grow them in an appropriate matrix like this Alvetex, which is the collagen comes out of the cells, and the cells remain more distant from each other. And this is how it looks like in vivo. So this is the epithelial cells on a real intestinal fold. Inside you have the stromal, which is made of fibroblasts, with the extracellular matrix and the single individual cells. So this is if you keep on growing these cells for a long time, you can go 2, 3, and 4 weeks and 5 weeks. This collagen stroma matures, gets more and more differentiated. You have a higher deposition of collagen and extracellular matrix. So the collagen is a proxy for all the components of the extracellular matrix. And as you can see, you can keep them happily 35 days without propagating them. Exactly for the reason that I said before. So if a cell is happy and begins to differentiate, it just stops dividing, or divides very slowly. So you have just a spontaneous change, and the cells do not overgrow. Why is important this? Because this is, again, a study that we did in vivo. So one important thing that we learn, and that basically everybody knows but shouldn't forget, is that if you want to replicate an organ in vitro, you should know as in detail as possible how it actually works in vivo. And despite we are working with intestine, which is not a new organ, so it's been widely studied everywhere. Actually, if you go out of the mouse and the human, the details of the molecular mechanism are not so well known. And in particular, we looked at this subpopulation of fibroblasts, which are called the telocytes, which form a network, as you can see here, under the epithelial cells of the rainbow trout intestinal epithelium, the rainbow trout, which is our experimental model. So there is a very continuous network, and there are molecules which are exchanged between this telocyte network through extracellular vesicles. And what we saw in our in vitro model, which made us very happy, is that we could see these EVs also in vitro. So this reinforced our idea that the enriched environment was providing a situation in which cells were behaving as much as possible as the in vivo counterpart. Indeed, if you have a look at these three models, so this is the classical transwell like this one. So this is the membrane, and this is the epithelial cells. So the epithelial cells sit on the membrane. They're quite happy. Again, they begin to slow down the proliferation, so you can keep them in culture for 2 or 3 weeks easily without the need of expanding them. But they maintain the monolayer, but they are generally flat, or in best conditions they're cubical. Whereas if you grow them in this combination with fibroblasts and epithelial cells, you see that the cell assume a much more columnar shape. And this is the real thing, so the rainbow trout intestine. So you can see that we are getting closer and closer to the reference model, which is our main aim. So they show a number of markers in which the enterocyte expresses the right molecules at the right place, and they look very much similar to their vivo counterparts. So we measure enzymes. So everything was looking very, very nice and very happy. Then we begin to use the models for actually using the model. So using the model for analyzing feed absorption. Again, I have no time to go in all the details, so I'll just tell you the conclusion of our work. And the conclusion of our work is that the complex platforms with the alveoles, with the fibroblast, with the nice-looking epithelium, first of all, it takes much longer to get ready for use. So it takes almost a month before you can actually use this platform. And if you are familiar with cell culture, keeping a culture up and running for at least a month, because then you have to use the platform, it requires a good degree of precision and good work. So it's definitely not for everyone to use it. Then there are very trivial but important technical issues. So the devices in which this membrane are sold within, basically they have an upper chamber which is smaller in its volume. So you need to have a certain volume in order to avoid continuous evaporation and concentration of the medium. So we need to have a volume which is just the limit of overflowing. And if you are trying to understand how things move from the upper chamber to the lower chamber, if there is an overflow around the margin, of course, that mess up all your data. And this was a limitation. And the same could be said by the fact that the thickness of this membrane limits the number of analyses that it can be done. Finally, since you have to keep the cell growing in vitro for several weeks, in the end, the membrane in a way sags, deformates, and again, it causes a communication between the upper chamber and the lower chamber, which make it useless for functional tests. So in the end, we opted for the simpler platform. And then we come to the actual use of this platform. So again, this is a summary of a very extensive set of experiments. But basically, in the end, we, together with all the other partners, the expert partners, in particular, Scratting, which is a big feed producer, they formulated 5 different feeds, and we had to compare them and end up to have a ranking of this different feeds, which would be more suitable for the best characteristic for rainbow trout feeding. And the good part of it is the fact that we use the same 5 diets, both in vitro with our system and in vivo with a traditional nutritional trial. So as I mentioned, you do the luminal digestion, you expose the feed pellet to the intestine enzyme, and so that they are transformed from pellet to something similar to what reaches the intestinal epithelium. And then you set up this barrier, and you can look at the morphological analysis, the integrity of the barrier, amino acid absorption, lipids absorption, enzymatic activity, a number of tests that have been developed along the project. This is just the most an example. So we basically one of the tests, one of the most easy but most indicative tests is to compare the integrity of the barrier with the absorption, the permeability to large molecules, which are not supposed to go through the barrier. So this indicates how the barrier is actually functional, and the 2 parameters goes together in the same direction. But sometimes some of the diets would damage the barrier, so you have a decrease of the electrical resistance and an increase of the flow of large molecules. So basically, we look at this, another parameter, and we were giving a score to these different diets. So basically, but the problem is, as I said, we really want to see if our data were meaningful for in vivo analysis. But the problem there is that you have to compare completely different sets of the different set of parameters. So in one case, you look at the old fish, you can look at digestibility, you can look at growth, you can look at how much feed you need to have a certain growth, and so on, together with some histological analysis. In vitro, you don't have all these parameters, but you, as I said, you have integrity of the barrier, absorption of small or large molecule, amino acid absorption, or fatty acid absorption, and histological analysis. So the 2 set of data are not directly comparable. But then we found out the most complicated things of all, that when you perform an in vivo trial, then unexpectedly, at least from my standpoint, that I'm sort of not so experienced in fish nutrition, but you have different answers. So it's not as if you have a different, a single grading of this diet, but you, depending what you're looking at. If you're looking at digestibility, if you're looking at somatic growth, if you're looking at food conversion, if you're looking at the health parameters. So the problem comes to the fact that instead of having a single ranking of the diet, you have at least 4. And so it's difficult which one you actually consider. So we somehow combine the various results. So if you have a combined average weighted average of the different values, you have this kind of degrees. So the best one was obviously the reference diet, the fish meal, and then there is this experimental diet, a single cell protein at 10%. So it doesn't really matter what there is, but this is a kind of ranking that you have. But if you look at in vivo health parameters, you have a slightly different degree, a slightly different ranking. So which one would our system predict? Well, depends, of course, even in our system. So if you look at the in vivo combined, this was what we obtained looking at you remember that we have 2 cell lines, one of the proximal and one of the distal cell line. So the proximal cell line gives a very similar ranking as the overall combined ranking that we obtain in vivo. If you look at the in vivo health, so as the health parameter, you have exactly the same ranking using the distal cell line. So even in vitro, depending on what you're using, depending on the test you are obtaining, you have different questions, different answers, and this corresponds to different physiological parameters. So not too bad after all. So which is the conclusion so far, is that the platform can provide a high resolution ranking of the different diets, so you can actually classify them with different values. These results, I haven't gone into the detail, but it's very important, of course, the repeatability of the result, and we went through a lot of effort in setting up protocols that are actually repeatable within the laboratories. And the other thing, which I think in the end is a positive answer, is that depending on the experiment you perform with the cells, you can have different ranking. And basically, the difficult part now is understanding how this different ranking correspond to the in vivo results. And this can only be obtained with a constant, with a numerous repetition of what we did. So comparing side to side in vivo results with in vitro results, and which I think is a general rule altogether. So it's very nice to have, it's very easy to have, or relatively easy to have nice in vitro models that look good, but this doesn't mean anything until you directly compare the same test in vivo and in vitro and actually go to verify whether these results are functionally meaningful. I spoke too much, so basically just to briefly mention that our university, as many universities, are pushing us to try to have some practical application of our results. So we went through a path leading us to this sort of thing. So we have come up with this proof of concept idea. So we're trying to see if NutraSeed, so the nutritional trial simulator, can be any good. So we repeat several times the kind of thing. So we test complete diets or single components of the diets, which maybe is easier for the in vitro system. And we'll keep on saying if looking if these results are actually of good quality. And finally, I would like to thank all the group, of course. Nicole is head of the lab. She developed the whole system every day in the lab. My colleague Tiziana Brevini, who is an expert in 3D modeling and interaction between the environment and cell differentiation. And Gaia and Deborah, who are newcomers in the lab, and they've been trained in all these techniques, and they can happily grow cell line for more than a month without contaminating them and taking out nice results. And of course, our sponsor. Thank you very much for your time.
Yeah, we can have one quick question. Do you guys have any questions? There.
Hello, Fulvio. Thank you very much. I was curious to know, thank you for a nice talk. Curious to know if this matrix that you prepare with fibroblast, did you try it with stem cells on the fish intestine?
Thank you. Thank you for your question. Yeah, as I said, I had to quickly summarize. So basically, when we grow cells on plastics, they grow very fast. And then, so I skip a long part of our job that was to identify rainbow trout intestinal stem cell, which is it's not so trivial. So it doesn't translate automatically from what is known in the mouse. So basically, to make it short, is that when you grow these cells in the plastic, stem cell constitute 30–40% of the whole population, and in fact the cell keep on growing. So we have to split them once a week because in general fish cells are slower than mammalian cells. They are grown at 20 degrees and so on. But when we move them in what we call the enriched environment, the stem cell population decreases, and because whereas the differentiated cells increase, so they differentiate given the environmental stimuli. They progress towards the differentiation tool. So we don't have a pure stem cell population because it would be too complicated to get, and I don't know how functional it would be. But in practical terms, that's what we got. So when we grow cells on plastic flask, the stem cell population constitutes a sizable amount of these cells, so to keep the population growing. But if you move them in a more stimulating, in a more differentiating environment, they move towards a more mature phenotype. And as I said, you can play around with the environment and get a more or less effective differentiation. What we found is that at least in our condition at present, moving towards the best differentiation in the end doesn't make any difference in terms of being able of grading the diets. Because then you also have to balance the complication of the growing technique with the effectiveness of the result that you obtain. So at present, we settled down with a not so sophisticated method, but still provide us enough sensitivity to grade the various diets. But we are working on it and trying to see if this can be improved and if the comparison can be made more effective.
Thank you.
Thank you very much.